Okay, so we left off with the electron transport chain, so in chemiosmosis. So if we look here, remember the two electrons from NADH go through the electron transport chain. Series of redox reactions provides the energy to pump the H pluses from low to high concentration. So 1, 2, 3 H pluses for every 2 electrons for every 1 NADH. And so therefore 1, 2, 3 ATP can be made from 1 NADH. And then for FADH2, drops off here and only goes through these two protein complexes before it meets oxygen and forms water, and only 2 H pluses are pumped, so 2 ATPs are made. So these as the H pluses diffuse back in, changes the shape of the ATP synthase, allowing ADP and your phosphate to bind, which are your substrates, and you get your product ATP, and we make many of that. And so oxidative phosphorylation, see the brackets here, electron transport chain, chemiosmosis, together make oxidative phosphorylation. All right, so we can put here your total. So how many NADHs are coming from the previous steps? So how many NADHs are coming? Go back and look, or can you remember how many NADHs from glycolysis, link, and Krebs cycle or citric acid cycle? So pause this if you can do that. All right, if you paused it, you should have come up with 10. 10 NADHs make, each one of those make 3 ATPs, so we get 30 ATP made from NADH. Now do the same thing for FADH2. Pause it for that. And you should have gotten 2 from the Krebs cycle. The 10, by the way, comes from 2 from glycolysis, 2 from the link cycle, or link reaction, and 6 from the Krebs cycle. And so therefore that's how we get our 30. And 2 from FADH2s come from the citric acid or Krebs cycle, and each one of those make 2 ATP, so we get 4. So our total is 34 ATP made in just this oxidative phosphorylation step. All right, so then see if you can pause this and figure out and do these blanks here. See if you can do that. So pause it. All right, so during cellular respiration, most energy flows in this sequence. So it starts off, you should have glucose. It's the original energy source. Now remember then, the first few steps of cellular respiration is to break down glucose and strip its electrons. And so then the energy at the end of the Krebs cycle is all in NADH and FADH2, your electron carriers. And then those electrons, then the energy is now in the electron transport chain. And then it says proton motive force. That's the when the electrons go through the electron transport chain, you get the H pluses pumped. H pluses are a proton, essentially. And so you have this force that can do work by diffusing back down through ATP synthase, changing the shape of this enzyme and generating ATP. So now our energy is in ATP. And I wouldn't expect you to know this, but about 40% of the energy in a glucose molecule is actually transferred to ATP during cellular respiration. The rest is released as heat. All right, so remember increasing the entropy and so on. So it's not 100% efficient. All the energy in the glucose doesn't end up in the ATP molecule. And altogether, what did you put here for the ATP? 34 of them were made in oxidative phosphorylation, but then you should also have 2 in glycolysis and 2 in the Krebs cycle, so therefore making about 38 ATP. All right, and that is cellular respiration, folks. But now we're going to take a look at something else. And look at this overview picture here. It pulls it all together for you. So when we look at this overview, showing you glycolysis broken down into 2-pyruvate, generating 2 ATPs. Notice by substrate level phosphorylation, that means the phosphates to hook onto ADP come from a molecule and generates 2 NADHs. And so then those NADHs are used in that third step. Here's the link reaction, 2 pyruvates to 2 acetyl-CoAs, Two NADHs go to oxidative phosphorylation. Citric acid cycle, the two acetyl-CoAs enter. You get some ATP too by substrate level phosphorylation, meaning the phosphates come off of a molecule to make ADP. And you get 6 NADHs and 2 FADH2s that go to oxidative phosphorylation. And all these electron carriers are used to generate we just talked about 34 ATP. Notice here about 32 or 34. It's still up in the air as far as exactly how many ATP are made by the H pluses diffusing back into the matrix. It says, by oxidative phosphorylation, depending on which shuttle transports electrons when it's from, I should say from instead of form, NADH in the cytosol. So notice here that NADH here, it says electron shuttles span the membrane. And so we have, notice here as it goes across, NADH or FADH2. What actually happens is that the mitochondrial membrane is actually impermeable. It's not permeable to NADH. So what happens is the two electrons that are being carried by NADH come over here, and those shuttle across the membrane, but NADH actually doesn't. So those two electrons, when they move across the membrane, can either be picked up by NAD plus and become NADH in the mitochondrial matrix, or by FAD and become FADH2. So depending upon which one it actually becomes, when these guys go to the mitochondria, what happens is, remember that the NADH, going back over here, makes one more ATP, 3 ATP, versus if it's those electrons actually become an FADH2 molecule, it enters the electron transport chain at a lower energy site, and so therefore only makes 2 ATP. So therefore, if those 2 NADH molecules coming from glycolysis stay as NADH, you get your maximum of 34. But if those 2 electrons get transferred to FAD and become FADH2, instead of these 2 molecules starting at the beginning of the electron transport chain, they're going to start here and make therefore 2 less ATP, and so therefore we get 32. So that's what it means by depending upon which way it is shuttled across the membrane. So that's why we get our end product 36 or 38. And quite honestly, if you look in different textbooks, that number will be slightly different. And so we're going to go with what our textbook goes with, but just know that in real life, the actual totals are debated, because I think that the amount of ATP made here is not a whole number. Like when I said that NADH makes 3 ATP, they actually think it's actually 2.5, and so on. So we get some logistics in there that for this class is not really that important. Okay? All right, and so then the last thing that we're going to look at is what happens when there's no oxygen present. So fermentation enables some cells to produce ATP without the use of oxygen. So we know that cellular respiration requires oxygen to produce ATP. Why? Oxygen is the final electron acceptor, right? Glycolysis can produce ATP with or without oxygen. Anaerobic, that's with oxygen, with O2. Anaerobic, without O2. So glycolysis happens no matter what. All right, so no matter if we're not breathing, we get a little bit of ATP. It's not enough to keep us alive. That's why if we don't breathe for very long, we die, or one of the reasons that we die. In the absence of oxygen, glycolysis couples with fermentation to produce ATP. So there are 2 types of fermentation that we need to be aware of. Fermentation consists of glycolysis plus reactions that regenerate NAD+, which can be reused by glycolysis. 2 common types are alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvate is converted to, what is it converted to? It's a type of alcohol. Alcohols end in OL, right? Ethanol. Converted to ethanol in 2 steps. The first, releasing CO2. When it says alcohol fermentation by yeast is used in brewing, winemaking, and baking. So fermentation occurs with glycolysis in the absence of oxygen. So we brew beer in an anaerobic condition. So let's look at this picture here. All right, so this part right here, I'm going to put a dot here. This is glycolysis. So down here is the fermentation part. So what happens in glycolysis? Let's review. Glucose is broken down into 2-pyruvate, and we get 2 ATP made in the process. So that's the 2 ATP in glycolysis. And then, remember, 2 NAD pluses are converted to 2 NADH. So that's the little arrow here. All right, and we make NADH. Now, usually, where does this NADH go? This NADH usually doesn't stay build up in the cytoplasm. It usually goes to the take the electrons to the electron transport chain, right? All right, but what happens is without oxygen, if I draw here the mitochondria, here's the mitochondria, and this is oxidative phosphorylation occurring here. So this is the ETC, right? And chemiosmosis. This is where, I'll just abbreviate there, this is where most of your ATP is made. Without oxygen, only glycolysis occurs. So we're only getting glucose getting converted to the 2-pyruvate. And guess what? Nothing else happens. So none of this happens here. And so what happens, remember, is that NAD pluses were used to make NADHs. Plus the ATP. I'm just going to draw this right now because that's what's important here. Do you see that we have a finite, if this is the cytoplasm, you have a number of NAD pluses in the cytoplasm. So here are your NAD pluses. And so what happens is when the glucose, normally if there's oxygen present, glucose is broken down into 2-pyruvate, making 2, I should write here 2 NADHs, and those usually take the electrons to the electron transport chain. When NADHs drop it off, it becomes NAD plus again, and can come over here and get more electrons and become NADH, drop them off at the electron transport chain, become NAD plus, and so on and so forth. So we're constantly shuttling these guys around, and it's becoming NAD plus, then NADH, NAD plus, NADH. That's if oxygen is present. This can only go into the mitochondria in the presence of oxygen, so with oxygen. So this, what's happening here with no oxygen, is this part is not happening, which means then oxidative phosphorylation isn't happening, which means that we're going to get a buildup of NADHs, and we're going to run out of NAD+. Can you see that? All right. One glucose molecule comes in, gets broken down with 2 pyruvate, and we make some ATP, 2 ATPs from 2 ADPs. 2 ATPs are made, cell stays alive a little bit, 2 NADHs are made, the cell's going to use up the ATP, they're not going to use the NADH because it's not going to be used in the electron transport chain. So you're going to get a buildup of this. But then what happens with the next glucose? The next glucose comes in, gets broken down into 2 pyruvate, you make 2 ATP, and it's going to use up another NAD+, and that NAD+ is going to become NADH, and then that's it. All right, nothing else is going to happen. Then the next glucose comes in, it's broken down into 2 pyruvate, we make 2 ATP, but guess what? We use up another NAD+ to make NADH, but then the NADH aren't used. The NADH are never reconverted back to NAD+. That process isn't happening, which usually happens in that third step. So what we're going to end up happening is we're going to run out of this NAD+. If we run out of NAD+, then we're really in trouble because we can't even do glycolysis anymore because you have to have the NAD+ there. So in comes fermentation. So this is what we have here. Fermentation. At the end of glycolysis, we get without oxygen, we get a little bit of energy, we get NADH made, and that's what we want, is a little bit of energy. So that we don't run out of NAD plus and can continue to do glycolysis, is the cells do fermentation. So in yeast cells, they convert the pyruvate to acetaldehyde. Notice here it's 2 carbons. This is 3 carbons, and so it loses carbon dioxide. So carbon dioxide is lost, and the acetaldehyde is then converted to ethanol. That's your alcohol. Right here. And in the process of converting acetaldehyde into ethanol, that NADH is converted back to NAD+, and now it takes the place of what should have happened in oxidative phosphorylation, where it drops off the electrons. NADH drops off the electrons at the electron transport chain, but that's not going to happen because there's no oxygen, so therefore we regenerate the NAD+. So now the next glucose molecule comes in, and we'll always have NAD+ to make glycolysis happen, to get a little bit of ATP with no oxygen present. So let's write that down. Fermentation replenishes NAD+, so that glycolysis can continue to occur with no O2 present. So replenish that up. And so alcohol fermentation happens in yeast. And so think about this. They put the yeast in an anaerobic condition, making it so that they can't do the full cellular respiration. They can only do glycolysis. So then they go through glycolysis, but to prevent the NAD plus from running out, they go through, after glycolysis, alcohol fermentation. And so the end product is alcohol, and therefore that's the alcohol in your beer, and then it produces CO2, that's the carbonation in your beer. And so therefore, the purpose for yeast is just to replenish that NAD+. Alcohol is just a waste product. It's a side thing that happens that we happen to use, and it's a billion-dollar business, billions of dollars of business. And so we happen to use that and take advantage of that, but the purpose for yeast is to regenerate that NAD+, so that when there's no oxygen present, they can continue to do glycolysis. So if we look here, we do not do alcohol fermentation. So in the absence of oxygen, we do not get a buildup of alcohol. We do what's called lactic acid fermentation. Pyruvate is reduced to NADH, forming lactate or lactic acid. It's again the difference between, is the carboxyl group intact or did it release an hydrogen? All right, so lactate versus lactic acid. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. In humans, our human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. So when we look at this one, here again, we have glycolysis and fermentation. So again, glucose to pyruvate generates 2 ATP, and we take 2 NAD pluses and convert it to NADH. But just like up here, the 2 pyruvates can't go in, and none of the rest of cellular respiration is going to happen. So the NADHs are not going to drop off electrons in the electron transport chain without oxygen present. And so therefore, we have fermentation, and our pyruvate gets converted in our muscle cells to lactate. Notice here, it has this is the carboxyl group. It has already acted as an acid by giving off the H+. If it had H and that was OH here, it would be lactic acid. So therefore, it's kind of like the same molecule depending upon if it's given off the hydrogen or not. And so in that process, it regenerates NAD+, so our muscle cells never actually run out of NAD+ and can continue to do glycolysis until we have enough oxygen to do the whole cellular respiration process, until we can do the whole thing. So our muscle cells, we cannot live for very long in this anaerobic condition. And also I want to point out, when you are working out, so if I sat here and squeezed, if you've taken anatomy, you've had done where you've squeezed a tennis ball or a ball like a tennis ball, and you've used your muscle really, really hard as fast as you can, and what happens is you start to get a muscle cramp. And so what actually happens is that in that situation, I'm still breathing as I'm using my muscles in my hand, squeezing this ball. Pretend like there's a ball. I'm still breathing. My body's still getting oxygen, but I can't deliver enough oxygen from my lungs through my arms, through the blood vessels in my arms, to get to the muscles in my hand to fast enough. And so what I've done is, by using this muscle so fast and I can't deliver enough oxygen to it, I've created a temporary anaerobic condition where my muscles aren't getting enough oxygen to do the full cellular respiration process. So so what happens is my muscle cells start to go through lactic acid fermentation, and this lactic acid buildup in your muscle cells actually creates pain. So then my muscles, it gets painful. So what happens when it gets painful? I actually stop. So I stop using my muscle. That's a protection measurement. So now my body can get enough oxygen to my muscles, and therefore, and your body will eventually break down the lactic acid, and therefore I'll go back to an aerobic condition where now I will start to do the full-fledged cellular respiration, even getting to oxidative phosphorylation, so I can get the maximum ATP. And so that's what's happening with lactic acid, and it causes cramping and pain and so on. So it's a protection measurement, and so then you'll create a situation where you get enough oxygen and get your maximum ATP there. And those are your two kinds of fermentation in either yeast or in our muscle cells, or also in bacteria. So let's look at how fermentation and cellular respiration compare. So both processes use glycolysis to oxidize, break down, and strip electrons. Oxidizing is losing electrons, so glucose is losing electrons, and other organic fuels. So we talked a little bit about how we can use fat and proteins. The processes have different final, and it should be, what do you think? Final electron acceptors. An organic molecule such as pyruvate in fermentation. So this is the final electron acceptor, is pyruvate. And then what's the final electron acceptor in cellular respiration? You should have O2. Cellular respiration produces much more ATP than fermentation. Why? Because in cellular respiration, you can do all the steps. When fermentation without oxygen, you can only do glycolysis, and for every glucose molecule, you can only make 2 ATP versus up to 38 ATP if you do the full cellular respiration process. So yeast and bacteria are what we call facultative anaerobes, meaning that they can survive using either fermentation or cellular respiration. So they can actually survive if there's no oxygen and survive off of just the 2 ATP for glycolysis and fermentation, or they can survive with oxygen where they can do cellular respiration. We can only survive in an anaerobic condition. So in a facultative anaerobe, pyruvate is a fork in the metabolic road that leads to 2 alternative catabolic routes. So what does that mean, a fork in the road? So let's look at here. Here's glucose to pyruvate. That's glycolysis. That happens no matter what. If oxygen is present, pyruvate goes and does the link reaction, gets converted to 2-acetyl-CoA, which enters the citric acid cycle, which then also goes and what is it going to do? Do oxidative phosphorylation. The electron carriers are going to do A. So you get a max of 38 ATP per glucose. If there's no oxygen present, you can only do glycolysis, and either pyruvate's going to generate and be converted to either ethanol in alcohol fermentation or lactate or lactic acid in lactic acid fermentation. And so here, what's our max number of glucose? It's a max of 2 ATP per glucose molecule. Why? Because you're just doing glycolysis, and glycolysis only makes 2 ATPs. And so therefore, that's your max. Big difference here. Big difference in the number of ATPs. So the evolutionary significance of glycolysis, it occurs in all organisms. They think that glycolysis probably evolved in ancient prokaryotes. Prokaryotes are like bacteria. Actually, before there was oxygen in the atmosphere, because it doesn't need oxygen. And the last concept we already talked about a little bit in the packet, but glycolysis and citric acid connect to many other metabolic pathways. So let's look at this. Glycolysis and the citric acid cycle are major intersections to various catabolic and anabolic pathways. Catabolic breaking down, building. So let's look at this. The versatility of catabolism. So catabolic pathways funnel electrons from many kinds of organic molecules into cellular respiration. We've just talked about glucose, but we've also talked about protein and fats. So glycolysis accepts a wide range of carbohydrates. It's not just glucose, although glucose is the main one. Proteins must be digested into amino acids. And amino acids can feed glycolysis or the citric acid cycle. So we talked about that when we went over that packet. So proteins can be broken down into amino acids, which can be converted to pyruvate and enter here, acetyl-CoA, and enter the citric acid cycle. This, remember, was the waste product. This is going to be urea. This is the waste. The ammonia that's produced that we talked about, that's not part of that's not part of cellular respiration. Fats are digested to, and see if you can think of what they're digested to. See if you can fill it in and pause it. All right, we should have glycerol and fatty acids. And so we see here glycerol enters into the glycolysis. Fatty acids converted to acetyl-CoA, make lots of acetyl-CoA. We talked about that already. And an oxidized gram of fat, meaning when we break down fat, an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. So way more energy in this. And then pulling in from last chapter here, biosynthesis. We also use some of these to actually build molecules. That's the anabolic pathway. So the body also uses some of these small molecules to build other substances. So we don't only just break them down, like all of our sugars and all of our amino acids we don't break down, we use them to build proteins and other carbohydrates. These small molecules may come directly from food, from glycolysis, or from the citric acid cycle. So that's biosynthesis, making living things or making molecules that are going to be part of living things. And then we regulate, we talked a little bit about this in Chapter 8 with enzymes, but we regulate cellular respiration via feedback mechanisms. So feedback, what's called feedback inhibition, is the most common mechanism for control. If ATP concentration begins to drop, respiration speeds up. We need more ATP if we don't, if we're using it all up. So this means using ATP. When there's plenty of ATP, what happens to respiration? It needs to slow down. So the control of this is based mainly on regulating the activity of the enzymes that work to catalyze these reactions, strategic points in the catabolic pathway. So the last picture here. So what we see is if we have an increase in ATP concentration, let's look here. This is glucose to pyruvate, that's glycolysis, link reaction, pyruvate to acetyl-CoA. Acetyl-CoA enters the citric acid cycle, and oxidative phosphorylation happens after that. These guys all make ATP. So if ATP starts to build up, you have a high concentration, you're not using it, let's say you slow down, then ATP actually inhibits this enzyme here. And if this enzyme gets inhibited, nothing beyond that's happening, and so we stop making ATP. Another thing, it says AMP. If AMP, the concentration increases. Let's talk about what AMP is. If we use our ATP, ATP breaks down into ADP plus an inorganic phosphate. But what happens is if your body is not, if you're using up ATP and you're not producing it enough, ADP can break down into AMP, which is adenosine monophosphate, and we can get a little bit more energy in it. So what happens is if AMP builds up, that means we're in dire straits, man. We need some ATP. So therefore, AMP stimulates and acts as an activator of that enzyme. All right, and so therefore it acts as an activator there and makes that enzyme work. And so we have an activator and we have an inhibitor to make that enzyme work, and therefore either cause cellular respiration to happen or not cause cellular respiration to happen. And then if you also get a buildup of citrate here, it also inhibits that. If you get a buildup of that, it also inhibits that enzyme. So we have two inhibitors in the stimulator, and that's how we can control and make this enzyme at the beginning turn on and off or be an active or not active depending upon these end products and so on. And that's how we regulate cellular respiration. All right, we'll pick this up tomorrow.